I would like to introduce our webinar speaker, Dr. Kirsten Heiss. Kirsten received her PhD in infectious diseases from the Ruprecht Karls University in Heidelberg in 2006, working on the malaria-causing agent Plasmodium. Further postdoc studies at the Institute of Immunology at the University Hospital of Hamburg-Eppendorf enabled her to combine infections with immunology. Back in Heidelberg in 2009, working as scientist at the Center for Infectious Diseases at the University Hospital Heidelberg, her research interest focused on vaccine development and the identification of novel targets for intervention strategies against malaria. In 2018, she joined the R&D unit of Pepperprint and is responsible for the discovery of novel antibody-mediated biomarkers in the field of autoimmune and infectious diseases. The title of her presentation is Uncovering Antibody Epitope Signatures in COVID-19 Patients by High-Density Peptide Microarray Screening. So once again, please use the question pane to type your questions if you have any. And we will also share these slides and a post-webinar file. So Kirsten, I'm handing the cursor over to you. Please take it away.
Yeah, so thanks, Kirsten, for this kind introduction, and welcome everybody to today's webinar dealing with peptide microarrays and their application in analyzing proteome-wide antibody responses in SARS-CoV-2 exposed individuals. So I would like to start my presentation with a few brief introductory words about COVID-19, although I imagine we are all familiar with it by now. So COVID-19 is an acute respiratory disease with a highly diverse clinical picture caused by the newly emerged coronavirus SARS-CoV-2. The map here on the left side shows the cumulative cases reported worldwide, with the dark blue color representing the highest number of confirmed cases. So as of yesterday, above 170 million cases are confirmed globally. A SARS-CoV-2 infection is possibly associated with long-term consequences, with persistence of symptoms or the development of new symptoms late in the course of the disease. And this is what we now know as long COVID. So here the symptoms are as heterogeneous as seen in acute COVID-19, and examples are chronic fatigue, numbness, shortness of breath, and also depressions are described. And it's estimated that approximately 10 to 20% of infected individuals are affected by this long COVID consequences. So strikingly, in a short time period, efficacious vaccines became available and vaccination campaigns are running globally. So however, we have to keep in mind that multiple SARS-CoV-2 variants are circulating and potentially new variants will emerge, which is a challenge, a huge challenge for vaccine development. Well, when new pathogens emerge, as we have the situation now, but it accounts also for all pathogens, understanding the host immune defense mechanisms and identifying the pathogens' Achilles heel will ultimately guide the design of drugs and vaccines. So this graphic here illustrates important host pathogen interactions. For example, the host cell entry by SARS-CoV-2, which is of course an important target for intervention strategies. With respect to host immune defense mechanisms fighting against SARS-CoV-2, these are shown here on the right-hand side. So we have now emerging data that have demonstrated the importance of both, so the cellular as well as the humoral immunity. And the focus of today's webinar are studies of the humoral immune responses. So in this context, what do we know? So the generation of neutralizing antibodies are important to combat viral infection. So one could ask, so why is it important to understand humoral responses? So what is the significance to study humoral responses? So one important point are, of course, intervention strategies. So strategies for active and passive immunization, but also the development of new diagnostic, serological diagnostic markers, and also epidemiological studies. So a rising question when it comes to antibody, the analysis of antibody responses. So I'm pretty sure the list would be quite long. So I only highlighted a few of this in this cartoon here on the right side. So responses to be analyzed are, for example, so antibody responses to the escape variants, or what happens with the vaccine-induced antibody response over time when we talk about years after vaccination. What is also an important issue when it comes to diagnostic, for sure, is the cross-reactivity between related coronavirus. And we also may add to this list here possible mechanisms that are responsible for this long COVID event. So are, for example, antibodies involved or not? So when we go to the literature, so most studies focus on SARS-CoV-2 antibody responses against a single target protein. And here the most famous candidates are obviously the spike protein as well as the nucleocapsid. However, this reflects only a small fraction of the humoral immune response. So considering the entire proteome will certainly give a more detailed picture of humoral immune responses. So for this, we were able to quickly produce a full proteome peptide microarray for SARS-CoV-2 within about a month of its sequence publication. And this became possible with our unique platform technology, which I will present in a bit. So the SARS-CoV-2 protein microarray covers the entire proteome of SARS-CoV-2 translated into overlapping peptides. A high peptide-to-peptide overlap, and we are talking here from 15 mer peptides with an overlap, with a peptide-to-peptide overlap of 13 amino acids. This allows a high-resolution epitope analysis. So if you now have a closer look to this figure here below, here you can already see representative staining images from our study, which I will present in a few minutes. So here you can see images from SARS-CoV-2 naive individuals versus SARS-CoV-2 infected individuals. And the red spots indicate IgG responses, and the green spots indicate IgA responses. So we analyzed both antibody isotypes in parallel. Our microarray is framed by control spots. So what you have here is an epitope resolved view on the SARS-CoV-2 specific antibody responses across the entire proteome of the virus. So as I mentioned in the beginning, new SARS-CoV-2 variants are circulating, and because of this, we recently updated our SARS-CoV-2 proteome chip and added peptides carrying the respective mutation from different variants of concern. So the unique flexibility of our technology makes it possible to quickly produce peptide microarrays with any peptide content. And you can see on the next slide how this works. So we have a laser printer, which is illustrated here in this little image, that has 24 printing units, one for each amino acid, which prints peptides directly onto glass slides through a series of melting and coupling cycles, which are shown here in this cartoon. And finally, we can produce microarrays comprising linear or conformational peptides. Well, the benefits of our technology, as I already mentioned, are a unique flexibility in terms of peptide content, fast production times, and a high spot density. But now coming to our study, which we performed in close cooperation with the Charité in Berlin and partners from the Munich Clinic in Schwabing. And the results of this study were recently published in Frontiers in Immunology. So in this study, we followed a two-branched approach investigating antibody profiles. First of all, longitudinally in COVID-19 patients with mild disease symptoms. And for this analysis, the sera were collected at different time points after symptom onset. So we included a very early time point and then an early and late convalescent phase. In the second approach, we compared serum from patients with a mild COVID-19 disease progression versus patients with a severe COVID-19 disease outcome. We used as a control cohort, of course, SARS-CoV-2 naive individuals. And for the profiling, we analyzed IgG and IgA simultaneously. So in the next couple of slides, I'm going to present our data on the longitudinal analysis of the epitope-specific antibody responses. Before doing so, I would like to share with you the data on antibody responses against the spike domain, which was determined by ELISA, and which is shown here above, and the neutralizing antibody titers for the respective patient samples that we used for our longitudinal screening. So what you can see is that responses against the S1 domain, as shown here, as well as the neutralizing antibody titers were heterogeneous across the COVID-19 patients. So we had, when you have a closer look here at the neutralization titers, we have a broad range combining low to high titers. So this heterogeneous breadth of antibody responses across COVID-19 patients was also detected on epitope level and across the entire protein, which you can see here. So shown are heat maps where the peptides are displayed on the horizontal axis together with the SARS-CoV-2 protein assignment, and the individual patient sera on the vertical axis. We have on the left-hand side IgG, and on the right-hand side the profiles for IgA. The color coding corresponds to increasing fluorescence intensity, meaning the darker the color, the stronger the signal. So for IgG, we observed an increasing epitope-specific antibody responses in most instances toward later time points. So it's a rather stable immune response. However, when we have a look on IgA here, we observed a rather weak early response and followed by a peak before declining for most epitopes at the latest time point of analysis. So for IgA, we have a rather short-lived epitope-specific response across the proteome. Well, I can imagine that in this figure, it is difficult to really follow the epitope recognition pattern, and to follow it even better, we calculated for each of the peptide the percentage of responders in the corresponding patient cohort. May I put your attention first on here the legend. So what we did is we defined responder groups and color coded them. And again, the darker the color becomes, the higher the percentage of responders. Again, peptides are shown on the horizontal axis, and this time the percentage of responders in the corresponding cohort is shown on the vertical axis. So with this representation, you can nicely see the progression of epitope recognition over time. If we just pick one example, for IgG in the NSP15, you see maybe here an epitope that is increasing, so the recognition reactivity is increasing over time, with one next to it showing a really stable recognition pattern over the entire time point, and also epitope reactivity, the reactivities that decline to the latest time point. So with this data in our hand, we next determined the significant SARS-CoV-2 specific epitopes, comparing the reactivity profile of these COVID-19 patients to the naive controls. And here I show you the results for IgG. So we identified 9 peptides reaching statistical significance. These are now shown here in these violin plots, showing the individual antibody responses by these individual dots here. And shown is now here the normalized fluorescence intensity values against, on the x-axis, the different cohorts. So what you can see is that most of the epitopes could be assigned to proteins located in the ORF1 AB polyprotein, and I would like to highlight one of those, and this is here the first on the top to the left side. This is an NSP15 derived peptide, which is significant throughout the entire time span of analysis. So also we have a significant detection already very early, so week 1 post-symptom onset. So this is a potential serological marker for acute, early, and late convalescent SARS-CoV-2 infection. I also would like to highlight one additional epitope, and this is the last one here. So this is an epitope derived from the spike protein, and this epitope is located in the S1 domain in close proximity to the S1, S2 cleavage site. And interestingly, this sequence partially overlaps with published immunodominant epitopes. And here one study showed that serum antibody recognizing this overlapping peptide inhibit the cleavage by furin. So this might be a highly interesting candidate, but of course for the epitope we discovered here, whether or not this is involved in furin mediated or inhibition of furin mediated cleavage needs to be determined. So beside to our approach analyzing the longevity of antibody responses, we were curious to identify antibody reactive epitopes associated with different disease outcomes. So as the identification of correlates of COVID-19 disease severity is crucial for clinical management as well as intervention strategy. So for this, we compared the epitope signatures in patients with mild symptoms versus patients with a severe disease outcome. And severity here was defined by the need of admission to an intensive care unit. So before sharing the data on epitope level, I would like to show you the ELISA data again for the antibody responses against the spike S1 domain and the determined neutralization titers for the respective patient sample we used for this analysis. And this is shown in this slide. So again, we have a heterogeneous pattern of antibody responses towards spike 1 S1 domain here in mild cases of COVID-19. And this is also reflected in the heterogeneous or in the wide range of neutralization titer. If we compare here mild and severe disease outcomes, it's obvious that the severely ill individuals show a higher antibody response against the S1 spike domain. And this is in agreement with other published studies. So when we now move to the analysis on epitope level, considering the entire SARS-CoV-2 proteome, the picture is as follows. So our analysis reveals several significant differential epitopes for mild and severe disease outcomes, as shown here. So first of all, please pay your attention on the left-hand side. So these are showing the results in volcano plots, having on the x-axis the log-fold change of the intensity of each peptide versus on the y-axis the negative log 10 of the adjusted p-value. So in this representation, each dot represents one peptide, and the colored one, so either the purple one or the orange one for IgG and IgA, indicate epitopes that were statistically significant, whereas all the gray dots represent epitopes that did not reach any statistical significance. So what we see here is compared to mild disease, severe COVID-19 is associated with a higher number of significant epitopes for IgG as well as for IgA. And if we now have a closer look into the significant epitopes, and this you see now here on the right-hand side, which shows the individual antibody responses for the identified significant peptides in a heat map, together with the peptide sequences and the antigen assignment. So the upper bigger part, so these are all epitopes that are associated with severe COVID-19, which can then be divided into IgG-specific one. Then we have three candidates identified that are shared between IgG and IgA. And we have IgA-specific epitope that are associated with severe COVID-19. And here below, the lower part, here you can see the epitopes that are associated with mild COVID-19. So when we go here through this list, we identified epitopes originating from different SARS-CoV-2 proteins. If we have a closer look to the severe ones, several epitopes here that are associated with severe COVID-19 disease can be assigned to NSP3 as well as to NSP12. To now have a closer look, I selected the top hit candidates for IgG and IgA respectively, so peptides that are associated with severe COVID-19, and this is shown here in this figure. So we have here an epitope which is derived from NSP3, so it's an NSP3 derived peptide, which was the most powerful epitope to discriminate severe from mild COVID-19 disease for IgG. Interestingly, the top hit candidate in this case for IgA is also an NSP3 derived peptide. And if we go to the top hit for a shared epitope, so this was significantly recognized for IgG as well as for IgA. Here we have an epitope that is derived from the ORF3A protein. So to sum up our study, so our proteome-wide antibody screening identified linear B-cell epitopes, which are potentially applicable as serological markers for early and or late SARS-CoV-2 infection. And I want to bring your attention back to one example here with this NSP15 derived epitope, which was stably recognized throughout our longitudinal analysis. I also would like to point out this spike S1 epitope, which may interfere with furin-mediated cleavage. And we also identified in our screening several potential biomarkers that are able to discriminate severe from mild COVID-19 disease courses. For validation, which proves the suitability as serological markers, For example, with the larger patient cohorts, they are needed for further validation. So in the last couple of minutes, I would like to take the opportunity to share with you the broad spectrum of peptide microarray applications. So with respect to SARS-CoV-2, numerous customers used our SARS-CoV-2 related peptide microarrays to investigate epitope-specific antibody responses. And I summarized here a few examples, and if you're interested in more, please visit our webpage. So here I picked 2 studies, and this, which used our peptide microarrays to analyze vaccine-induced antibodies in the animal model. So we have a mouse study as well as an immunization study with chicken. And I also picked 2 further studies who analyzed the proteome-wide screening using patient serum samples. So of course, the research area of infectious disease is diverse with many different pathogens and applications. And this I would like to show you here. So we can produce peptide microarrays of any content. So bacterial, viral, or parasitic origin, which can be screened with sera from different origin or also purified antibody, depending on your research project, which can be then used for antibody profiling. So question in the area of vaccine development and monitoring, diagnostics and epitope mapping, of course, can be addressed. And to give you some examples here, I picked up a few for the field of vaccine development. So here we have an example from the parasites in the malaria vaccine development. Here the authors analyzed the epitope specificity of vaccine-induced antibodies after their successful clinical phase 1 trial. We have a viral example. I picked a viral example where the authors compared epitope signatures in vaccinated versus Ebola surviving individuals. For bacteria, here the authors used our arrays to identify immunogenic epitopes, which they then later used for vaccine design. And finally, an epitope mapping of a monoclonal antibody against CMV infections. So finally, also a few examples in the field of diagnostics. So I picked 2 examples from flaviviruses, where the authors, on the one hand, were interested in identification of discriminating markers between Zika and dengue infection, or also here discriminating markers for different clinical phenotypes. Last but not least, I added examples from parasitic worms for diagnostic purposes in humans, and also one for the veterinary medicine, because here this guy here infects domestic animals. So this was for veterinary medicine. So with this slide, I am almost at the end of my presentation. So the focus of today's webinar was indeed infectious diseases. However, peptide microarrays can be applied in different research areas. Other examples are autoimmune research and also cancer research. And with this, I would like to thank for joining the webinar, and I'm happy to take any questions.